In our explorations of genes and genetics and how they work, we want to talk a little bit about meiosis. And meiosis is something that we talked about in reproduction in terms of that's how sperm and eggs are produced, but really didn't have a great appreciation for the process. So here's the process. In your reading assignment, you looked at mitosis, so think about the steps of mitosis: prophase, metaphase, anaphase, and telophase. And in meiosis, we're also going to have those phases, and very, very similar things are going to occur. So here we have this cell. Let's say it's a stem cell for sperm or for eggs, doesn't matter. And it starts meiosis. So what's going to happen is in prophase, the chromosomes are going to coil up in bundles. The nuclear envelope is going to disintegrate, and that was just like in prophase of mitosis. In metaphase, the chromosomes line up along the middle of the cell, and in this case, those chromosomes are going to swap pieces and parts back and forth. So we started out with all blue chromosomes and all red chromosomes, and what we get here are part red, part blue in random mixes. So if you remember about reproduction, we said that the male human body makes about 100 million new sperm a day, and they're all genetically unique. So that means that the genetic uniqueness comes from crossing over. That's what this swapping back and forth of pieces is called. So if you're randomly swapping pieces and parts back and forth, and then randomly sorting out those individual chromosomes to separate cells, you'll see here in a minute we started with one particular cell, and we ended up with 4 genetically very different cells at the end of the process. And every time it does this, it does the crossing over in a different combination, totally random. So the odds are that you're going to get completely unique cells every time this happens. So in anaphase, the chromosomes pull apart half to one end of the cell and half to the other end. But just like in mitosis, we duplicated the chromosomes first before we started this. So we were diploid to start with, we duplicated the DNA, and then when we're pulling these parts back and forth here, each resulting cell at the end of this process in this image is still going to be diploid. So anaphase pulled them apart, half to one end, half to the other end, and we're hoping that we got exactly half of each type in each new cell. So we have 2 of chromosome 1, 2 of chromosome 3. We don't have only 1 of those or maybe 3 of those for one chromosome. That causes some serious problems. And then in telophase, you're going to reform chromosomes and then actually split that cell into 2 separate cells. So at that point that we have completed meiosis 1. But meiosis actually goes through these 4 steps 2 times. This is just the first step. And in this process, we went from 1 diploid cell to 2 diploid cells, and we had crossing over occur. So really the only difference between meiosis 1 and mitosis is the crossing over event that happens somewhere there in late prophase, early metaphase. With meiosis, we then go through meiosis 2. And if we're making sperm, we'll go right into that as soon as we finish meiosis 1. But if we're making eggs, we'll sort of pause and only complete that process after that particular egg has been ovulated. Or when we're getting closer to the ovulation event, this process will actually complete. So part of it's done early in the process, in the case of human females before birth, and then finishing up meiosis 2 happens after ovulation, so during reproductive ages. So in meiosis 2, we're going to take each one of those diploid cells that we made in meiosis 1, and we're going to go through prophase, metaphase, anaphase, and telophase again. But in this case, we are not going to do crossing over again. We already did that. But when we split, we haven't duplicated the DNA. So we're going to go from a diploid cell in prophase 2 to, by the end of the process, 2 haploid cells. So 2 of each chromosome to start meiosis 2, and by the time we finish meiosis 2, each cell is going to have 1 set. To make then a fully functional half of the DNA egg, or a fully functional half the total DNA sperm. Otherwise, the steps in these processes here are just like they were in meiosis 1. The only difference here is we're going from diploid to haploid at the end. So the result here was 4 haploid cells at the end of meiosis. So if we stack mitosis and meiosis next to each other, we're going to say that mitosis had 1 diploid cell that became 2 diploid cells. Meiosis had 1 diploid cell that became 4 haploid cells and engaged in crossing over in the first meiosis process. So here's that crossing over, looking at it a little closer. We've got these chromosomes and they're sister chromosomes. And what that means is that they are carrying the information for the same thing, and they were made, one of them was made from the other one. So it's really got the same kinds of information there. But one of them might have information for brown hair, one of them might have information for blonde hair. And so in this crossing back and forth, we might trade pieces and parts of those things back and forth, so that chromatid 1 might have a little bit of a new piece in that section, so calling for something a little different than it called for before of the same type. So hair color is not going to change to become an eye color thing, but a hair thing might change to a different hair thing based on which piece it picks up from which area. So here, our outcome of these guys sort of swapping back and forth is the first chromatid didn't change from its original condition. The second chromatid, though, it did pick up some pieces and parts. So basically, chromatid 2 and chromatid 3 traded some pieces. Chromatid 1 and chromatid 4 didn't change. That was just random. So any particular time you go through this process, any of those pieces in the proper spot, A's can swap with A's, B's can swap with B's, and that's a normal process of crossing over. If an A swaps for a B or a C swaps for a D or something like that, that's not normal crossing over. That's now considered a mutation. And we'll get to mutations in a little bit, but they almost never produce a desirable outcome. Every once in a while something good comes from a mutation, but for the most part mutations are considered at least potentially problematic. So we want A's to stay where they're supposed to be, B's to stay where they're supposed to be, on down the line. But we can swap A varieties back and forth, and that's okay. Hopefully that makes sense. But this process is really what gives us so many unique outcomes. So if we started with the cell that had the original components, mixed and matched them, and then split them in half so each resulting cell only has one of these chromatids, for example, we can actually have 4 cells that are each completely different than the original parent cell was for that particular chromosome. It's still phenomenal to imagine 100 million new genetic combinations every day, but this is the process that does it. So hopefully that sort of explains why it is a big deal, but it's not quite as big a deal as it sounded like it might be up front. So if we look at a sperm doing that, we go from that primary stem cell. Meiosis 1 gives us 2 diploid cells. Meiosis 2 splits each of those 2 diploids into 2 haploids, and then there's going to be some development. In this case, they're somewhat sperm-shaped, but they need to rearrange their shape a little bit. They need to streamline that great big floppy head they've got going on and develop a functional tail. So the metamorphosis and maturation part, the genetic chromosome numbers didn't change. It just sort of tweaked its shape and its physical structure so that it would be an effective swimmer. If we look at eggs, when we went from 1 egg to meiosis 1, then meiosis 2, and we're going to get 4 possible egg outcomes from this one original, but 3 of the 4 are going to be sort of tossed aside. So they didn't really become fully independent cells. They become these polar body structures that you see. So that's where we've divided into 2, and we're kind of pitching one over here. We're pitching another one off over here, getting rid of those extra chunks of DNA that could have resulted in 4 independent cells, but we really say the best one out of every 4 actually finishes. The other 3 scrap those, Let's not waste any effort on those. Because eggs aren't about quantity. Eggs are about quality. So pick the best one you've got to work with, move forward with that one, and don't even bother finishing making the others at all. In the case of sperm, it was about quantity. Because yes, we needed a reasonable quality, but we needed so much quantity that every single cell that is a potential to be made actually is made, and most of those have some potential at least of doing some sort of spermy thing. So here are those processes side by side, meiosis and mitosis. Some differences there, mostly in the crossing over for meiosis, in the 4 haploids instead of 2 diploids for the mitosis. But again, within a particular step, the same processes are occurring. Forming chromosomes, lining them up, separating them in half, dividing the cell in half. Those things work pretty much the same between both processes. Now here's an interesting example that happens in some characteristics that are found on the X chromosome. This is a calico cat, or a turquoise tortoiseshell cat. And with tortoiseshell or calico cats, they're always going to be female. So here's what's going on with that. Because females have 2 X chromosomes and males only have 1 X, there's the process known as X chromosome inactivation. Sometimes it's called dosage compensation, and that's a little bit more perhaps self-explanatory. Because the female has 2 X's, but the female doesn't need to express twice the amount of X chromosome expression, one of those 2 X's gets turned off in every cell. And it's kind of random chance in a way as to which one's turned on or turned off, at least to some degree, so that 1 X worth of DNA is expressed in a female to match the 1 X worth of DNA expressed in the male. In the world of cats, in some varieties of cats, their color gene is found on the X chromosome. So if a female has 2 X chromosomes, and the color possibilities, by the way, are black, orange, and white. So a chromosome might have the color of, let's say, black, or it might be an orange chromosome. We could say then white would be not calling for a particular color, just defaulting to white, let's say. And then that cat got, if it's a female, got 2 X's, one from daddy, one from mama. And depending on what those 2 X's contained, that will then determine the color possibility of this female cat here. So if the mama cat happened to get a black X from one parent and an orange X from the other parent, that cat has that potential to express either one based on which X chromosome is turned off and which X chromosome is turned on in any particular cell. So some areas of this cat, and the book refers to this as being a mosaic or a mixture of possibilities. In some areas of this cat, the X with the orange color gene is turned on and the black gene is turned off. And in other parts of the cat, the black chromosome is turned on and the orange one is turned off. So wherever the orange happens to be turned on, it's expressed. Wherever the black happens to be turned on, it gets expressed. And in areas where neither one of them perhaps was turned on or fully expressed, you get the white. So that gives you all 3 colors in the same cat. Because males are only going to have 1 X, they're either going to be black and white or orange and white. You're not going to find a male with all 3 because they only got 1 X, which means they only got 1 color option. They only got black or they only got orange. So that's why we can safely say that calico cats, true calico cats, are female, is because that's the only way they could get 2 non-white color genes, is to get 2 different types of X's, one from the daddy and one from the mama. Hopefully that makes sense. There are a lot of other things that can go wrong with chromosome things, and we wouldn't want to say the calico cat was something went wrong. That was just an unusual expression based on cat coloration patterns. The same idea can hold true for humans with other things as well. Humans can express pigmentation in a mosaic fashion too. It's not quite as common, but it does happen. Here's one, unfortunately, relatively common example of a mistake in chromosome number and arrangement, and this is called trisomy 21, or Down syndrome. So what we have is chromosome 21 here. Typically, again, we want to have 2 of each chromosome present in an offspring, and this one got 3 of chromosome 21. And this chromosome is a little bitty one. So it's got 3 little bitty chromosomes in this spot. It's only supposed to have 2. And so if you think about someone with Down syndrome, they have a lot of physical deformities. They have a lot of intellectual challenges. They have a decreased lifespan and increased health complications and problems. And there's a lot of social interaction and emotional challenges that go along with this. And all of those things because a little extra chunk of DNA here. So a little extra piece, it's little, but it causes a lot of problems. And so the reason why those didn't sort out was because there was a mistake in a gene in there, and it just caused the sorting process. When the cells were going through anaphase, they were supposed to be pulling apart. They didn't pull apart properly. In this case, it's probably going to be in the mama. Theoretically, it's possible in the daddy's genes, but probably in the mama's genes. What happened is that when you were supposed to be in anaphase and go from diploid to haploid, 2 of those chromosomes went one direction and none of them went the other. So instead of getting 1 and 1, you got 2 and 0. The one with 2 happened to be the egg that was ovulated. Then a sperm comes along with its 1, adds it to the system, and you got 3. Okay. Now, normally extra pieces of DNA are immediately fatal. So this is one of the relatively rare or very rare circumstances in which an extra amount of genetic material wasn't fatal, but it was very problematic. Now, the number one cause for this mistake occurring is old eggs. So a mama who is past the age of 35. Now, I know we probably don't consider a 36-year-old female to be old, but in a genetics world they are, because remember, those eggs have been sitting there since before that baby was born. So that mama who is 36 years old has 36-year-old eggs she's trying to build a baby with. Now, would you ever expect to pull an egg off the shelf at the grocery store that's been sitting there for 36 years and expect it to still be good? The answer is no, you wouldn't. So it's therefore unreasonable to expect a cell that's been sitting here, hasn't even finished meiosis yet, but it's been sitting here for 36 years, to be pulled into service and expect everything to work properly and sort out and divide. No, you don't. So that's why it happens most commonly in mothers who are over the age of 35. So that's a good biological age cutoff. Have all your babies before the age of 35, and that significantly reduces the risk of Down syndrome and every other genetic disorder out there too. So probably biologically somewhere between the ages of 25 and 35. Let's back it up and say really between the ages of 25 and 30 are the best years from a biological perspective and a medical perspective, the best age range to have those babies. But we don't want to stack them too close together either, so if you need to run a little bit on the back end, that's okay. So finish it before 35, and your odds of Down syndrome go way down, and all those other problems too. Quite frankly, at a certain age, you're no longer physically up to the job of raising those kids, and if you have them too late in the process, you might be retired before your kid graduates high school, and that would be a potential problem. There are lots of other very, very specific problems. The book talks about a fair number of them. So make sure you read about Jacobs and Turner and Klinefelter and Poly X and things like that. Those are all some problems with chromosome number. If you get too much or too little of something, or you're missing a piece of this or that, then you can have those sorts of problems occur. Here are the most common ways in which mutations or mistakes in the DNA occur. And it's simply a misspelling. So the letters, the codes, they got put in the wrong order. In image A here we have a deletion. So it should be A, B, C, D, E, F, G, and somehow or another the little A piece got deleted, broken off, it didn't go where it was supposed to, and you have B, C, D, E, F, G, so you're missing a piece. And depending on what that particular gene was that you're missing as to what the outcome is. But again, it's not going to be a good thing, so that's a problem. A duplication occurs when you accidentally copied a piece that wasn't supposed to be copied. In this case, we ended up with A, B, C, D, E, D, E, F, G. So we had 2 DEs in there. And that was extra material. It gets in the way, it breaks up the proper flow, and that's a problem too. An inversion occurs. A, B, C, D, E, F, G becomes A, B, D, C, E, F, G. So a couple of pieces in there somewhere. And it doesn't have to be these particular letters either. It could be anywhere in that chromosome that it just got flipped around backwards. And any piece could have been deleted, any piece could have been duplicated. A translocation might occur when you're supposed to have one chromatid have one set of information and a chromatid next to it have another set of information, and it's almost like crossing over occurred between the two of these when it wasn't supposed to. And so that leads to certain genes being expressed in one place and not in another place. And this is a possible cause for some forms of mosaicism, not exactly like we saw in the tortoiseshell cat, but it could cause some things to get misplaced, put in the wrong place, the wrong order, and get the wrong outcome at the end. Some interesting things happen, though, in the world of plants. Plants aren't quite as rigid or problematic with these kinds of changes. A lot of times plants just sort of roll with it, and they roughly do their job. They just come up with some interesting outcomes. So most of the time when you see plants that have variegated leaves, so some parts are white, some parts are green, or if you have, let's say, an eggplant, a traditional eggplant fruit is a dark purple, but you might see some eggplant that are striped. They would have purple and white stripes. Or in the fall with ornamental Indian corn. That's probably some sort of racist or something to call it Indian corn, but being a significant portion Cherokee myself, I'm going to say it's okay. You have my permission to still call it that. Basically, you often end up with kernels of corn that any particular kernel might be a whole solid color, or you might have an individual kernel that's striped colors. And those are really, really interesting from a cosmetic perspective. And genetically, what's happening here is translocation. So the purple gene is jumping into places where it normally would have a white gene present, and so some parts of it express the white, some parts of it express the purple, and it makes for some really interesting cosmetic appearances. In that case, the plant didn't really care either way, and that seed is still a perfectly viable seed. It just has some blending of characteristics that produced interesting outcomes. So it turns out that as humans, we're not really flexible with genetic and chromosome changes and switching around. So if it switches around much, we just can't handle that. But plants and a fair number of other animals don't really mind so much, so you see that a lot more. Here's an interesting translocation type of event. We've had some segments here that had some crossing over, or were going to engage in crossing over, but we had some inversion occur here. C and E traded places. They shouldn't have done that, and so that put that particular chromosome in an unusual combination. And then when you engage in crossing over with it, you get some inversion, some duplication, some deletion, some everything going on, perhaps in both of those chromosomes after they engage in crossing over. So that particular problem that occurred only in one chromosome became ultimately a problem in both chromosomes after crossing over occurred. So with those sorts of things, they're probably not going to turn out alive at all, in a human at least. Again, plants and other things a little bit more flexible with that. The point here is there's a half a dozen different ways, at least, in which these mistakes can occur, and depending on what the mistake was, how big that kind of mistake was, and in what chromosome it occurred, that will determine the outcome. So the outcome can be minimally effective and problematic all the way to completely fatal, or anywhere in between. Hopefully that all made sense, and some of it can be really, really interesting. We could spend several weeks talking about all these sorts of things in great detail, but that's at least a brief overview of the processes there. So thanks for joining us, and we'll see you next time.